And so now for the final part of the third lecture, where we'll talk about the 3 crunches that you'll be doing. So in the first 2 crunches, we'll ask you to predict the effect of held out genetic perturbations on the adipocyte gene expression profiles, so on the gene expression profiles of the individual cells. And the difference between crunch 1 and 2 is that in crunch 1, you'll be using this single gene knockout data set that we spoke about, experiment 1, while in crunch 2, you'll be working with this double gene knockout experiment, what we introduced earlier as experiment 2. And what we'll be evaluating on is, first of all, how you perform on whole transcriptome predictions, so predicting the expression level. And in parallel, we'll also evaluate on the ability of your models to predict the proportion of cells expressing each of the programs that we've spoken about and are interested in, so the differentiation programs and some metabolic programs. And so we'll announce winners separately for each of these tasks and separately for each of these 2 crunches. So for crunch 3, we'll instead be asking you to propose new double perturbations, so 2 gene knockouts, that skew the proportions of specific adipocyte states. So for example, we'll be asking you what the top 2 gene knockouts are that your model predict will most push towards this differentiated adipocyte state. And initially, there won't be new data that will be generated for this crunch, but the winners of crunch 1 and the winners of crunch 2 will have their top predictions, their top double perturbation predictions experimentally validated, and this will be the set that everyone is evaluated against. So everyone should participate. Okay, so that's it for the introductory lecture series. Thank you for following along, and we can't wait to see what you create.